Welcome back to the lab, everyone. Today we're going to go over the urinary system. All right, so today let's look at the anatomy of the kidney, and I've got a model of the kidney on the right-hand side, and I have a specimen on the left-hand side. So hopefully I'm going to kind of combine these two today, and let's see if that helps you guys kind of bridge the gap between the model and the specimen. So before I like to dive into the specific structures, it's really important we know the geographical regions of the kidney. So here I've used tape. If you just look between the arrows, that'll make more sense of it. This outer portion right here is the renal cortex. So this whole portion right here is the renal cortex. And then as we move medially, this section right here is the renal medulla. Again, we are not talking about any specific structures yet. We're talking about this whole area. So again, renal cortex, and then we move in medially. This is the renal medulla. We go in medially again, and then we have all of these structures right here. So this whole portion, and really I'll go ahead and move that over. It's really all of this right here. So these structures, that is the area we call the renal sinus. And again, we're going to dive in and look at what these individual features are called, but the most medial portion inside the kidney is the renal sinus. We go medially again, and this indentation is the hilum or the hilus. And it doesn't matter which term you use. That term hilus just describes a general area where we're going to have blood vessels and or other structures that are going to kind of connect into an organ. So here on the kidney, this indented region, which you can't really see that well on the model because it's on the underside of it, that's where we would find the ureter, the renal vein, artery, and then the renal vein. On the specimen here, this would be the renal cortex. This region right here is the renal medulla. This region right here is the renal sinus. And then this is the hilum. And actually on the kidney, on the specimen rather, you could see how it kind of goes in right here. That is the renal hilum. Okay, the way I like to teach the kidney is through two ways. First is looking at blood flow, and then the second is looking at filtrate, tubular fluid, and urine formation. And it's important that we understand kind of where the two meet up. So here we're going to start medially, and then we'll work our way laterally or deeper into the kidney till we get to the capillary beds, and then we'll turn around and come straight back. So here, this is obviously the renal artery. From there, we go into the segmental arteries. From the segmental arteries, then we go along the sides of these renal pyramids here called interlobar arteries. And the reason why they're called interlobar is because interlobar means in between the lobes. So what is a lobe? Well, a lobe is going to be a renal medulla, part of the renal columns on either side, and then the renal cortex. So if we were to take little pie slices like this, that's a lobe, that's a lobe, that's a lobe. So then that word interlobar artery makes so much more sense, and I've got that labeled right here. Remember, because arteries go A for away from the heart, we're going away from the heart towards the capillary beds of whatever tissue or organ we're looking at. So from the interlobar artery, then we're going to go into these vessels that make an arc shape. So if we have an arc shape, which is actually at what you would think of the base of these pyramids, that is the arcuate artery. So interlobar to arcuate. From the arcuate arteries, then we're going to have these smaller vessels that go into the cortex. And those are called the cortical radiate arteries. There is also another term, interlobular, but I have found many times over the years that students mix up the terms interlobular and interlobar. So do yourselves a favor and just learn cortical radiate arteries, even though that's a little bit longer. The term is actually extremely visual. So if we think cortical refers to the cortex, radiate because these vessels are kind of radiating into that cortex. Once we get to this cortical radiate artery, we're going to have to zoom into the nephron. So looking here on the pig kidney, here we would have the renal artery and vein. We actually don't see that here. It's missing, but you can imagine if we had these large vessels, and let me see here, we have maybe a tiny portion of the vein, but they're gone. And so these are color-coded based on oxygenation. So the pinkish would represent a systemic artery, and the blues represent a systemic vein. The yellow, when you're talking about the urinary system, yellow makes you think of urine. And so when we get to that part of the discussion, I'll point those out. So this is where the artery and the vein would be. Obviously, I would not test that unless I actually had a vessel to put a pin in, but we've seen this several times on the model, so you guys can put that together. For the pig kidney, then the other major ones we'd want you to be able to recognize here, so I'll pick this up, are these interlobar vessels. We mainly see the veins. We see a little bit of the arteries, but we mostly see the veins. And then you can see how we kind of have these little arcs right here. Those are the arcuate vessels. Again, we see more blue, so we're looking at the veins. And then the interlobular or those cortical radiate vessels, those are in the cortex. Remember, cortical radiate, these vessels are radiating into the cortex. Let's zoom in now, just to give you some perspective. I've got a lot of labels here, but we'll make sense of them all. We have taken this section right here and we've blown it up. So this right here is the renal medulla. Okay, this is part of a pyramid. And then this area right here is the renal cortex. So just a little bit of backing up. Remember, in between the lobes, so on the sides of the pyramids, we're going to have these interlobar vessels. So continuing with the arteries, we'll have that interlobar artery, and then this arcuate artery makes an arc. It also creates the border between the cortex and the medulla, so that might be another way you can remember those two. And then from the arcuate artery, let me just move this label for now. These are the cortical radiate arteries. So this is going to radiate into the cortex. So that is our cortical radiate artery. From there, we're going to have these very small branches. So this afferent arteriole, remember that the suffix I-O-L-E, eole, kind of reminds us we're looking at a smaller version of something. So whereas we had the bronchioles versus the bronchus in the respiratory system, here we have an arteriole versus an artery. So it's a very small artery. That afferent, and it's really afferent, but I like to overemphasize the first letter when I'm teaching it so you guys know exactly what I'm referring to. This afferent arteriole is going to go into this structure here called the renal corpuscle. So we call this whole structure the renal corpuscle. And I'll come back to that in a minute. This ball of capillaries we see inside here, that is the glomerulus. So remember that the term capillary just refers to the smallest blood vessels we have. We mostly think of gas exchange at the capillary level, but that's actually that's not what's happening within the glomerulus. So just think if this vessel is going into this small ball of capillaries here called the glomerulus. It's the afferent arteriole. I'm going to move to this one right here because it's we have a better example of it. So from the cortical radiate artery, we will go into this ball-like structure called the renal corpuscle. So that right there is the afferent arteriole. The vessel, let me kind of modify my sticker a little bit here. The vessel that is going to be exiting. So when I say it is exiting, that is the efferent arteriole. Again, you would actually pronounce this efferent. So it's afferent and efferent, but those sound very similar. So I like to mispronounce it on purpose. So I think, and it also helps you remember it. So I think E for exit, E for efferent. This is exiting that ball-like structure. So this vessel, the efferent or the efferent arteriole, is then going to connect to a capillary bed. And in the kidney, we have two kinds of capillary beds. We have the peritubular capillaries. It's because this is the network that surrounds the tubules, which we'll talk about in a minute. And then we have the vasa recta. And the main difference is that the peritubular capillaries are more so located in the cortex versus the vasa recta, which are more so located in the medulla. This is where gas exchange occurs. Same here with the vasa recta. So from these peritubular capillaries, when you guys have been doing your trace assignments and you're thinking, okay, we're going from, the renal artery to the capillary beds of the kidney, we were very big picture there. This is what we've been talking about, this spot here. And now we're just learning all the other little vessels that were in between the renal artery and the capillary beds and then back to the renal vein. So on our way back, this is where we do a U-turn. So from here, we're going to connect to the cortical radiate vein. That cortical radiate vein will then connect to the arcuate vein. And then from the arcuate vein, we will go to that interlobar vein. So let's zoom back out to the big model so we can see how all of this connects to that renal vein. Okay, so let's look at the venous return. So we've zoomed back out to the gross anatomy. Here we're at the cortical radiate veins. And remember, a vein is going to return back towards the heart. So we're just going to go in reverse. So from the cortical radiate vein, then we're going to return to those arc-shaped vessels called the arcuate veins. And then the arcuate veins will then go into those interlobar veins. And we actually don't have any segmental veins. Those interlobar veins just drain directly into the renal vein. And remember, that renal vein is going to take us back to the inferior vena cava. On the specimen here, same thing. Kind of already went over this a minute ago, but it doesn't hurt to look at it more than once. Here we have these cortical radiate veins. Those will drain into the arcuate veins. The arcuate veins then go into the interlobar veins, and then the interlobar veins eventually come together and merge to form that renal vein, which we do not visualize well on this specimen. Here we might see a small portion of it, but that renal vein will again connect to that very large inferior vena cava. Let's talk about where blood flow and filtrate and tubular fluid and urine formation meet together. So on this section right here, I was telling you that when we see these ball-like structures, those are renal corpuscles. And so the renal corpuscle is going to be made up of this ball-like structure, which is what we call the glomerular capsule or Bowman's capsule, and what's inside, that small capillary bed called the glomerulus. So this is a zoomed in version of one of these ball-like structures. So again, the entire ball-like structure is called a renal corpuscle. The renal corpuscle is made up of the outer glomerular capsule or Bowman's capsule and then the inner glomerulus or that inner capillary bed. We can further break down the glomerular capsule into its outer parietal layer, so that's this specific part right here, and then the inner visceral layer. So the visceral layer is going to be made up of these structures called podocytes, and so that's what's represented by these right here. And then in between the visceral and the parietal layers is the capsular space. Over here, we have the afferent arteriole and then the efferent arteriole. And how can you tell the difference? Well, the afferent arteriole is going to be the larger vessel. And there are some specialized cells on the afferent arteriole that are part of the juxtaglomerular apparatus, which we'll look at in just a minute, that also tell you that this is for sure the afferent arteriole. So the big picture is that blood is going to come in through this afferent arteriole, go through this glomerular capsular bed, and then we will have things filtering out of this capillary bed into the capsular space. So there's fluid in here, and things like water, glucose, amino acids, some hormones or vitamins, and then ketones can actually come through these little fenestrations, these little slits. And when it comes into this capsular space, that is eventually, farther down the line, going to constitute what we know as urine. So from the capsular space, once that stuff has filtered there, it will go into the proximal convoluted tubule, which I've abbreviated as PCT, but make sure you always spell things out. Just to recap what we said, blood is going to come into the afferent arteriole. Things will filter out, and then whatever stays in the capillary bed will exit via the efferent arteriole and head towards the peritubular and or the vasa recta capillary beds of the kidney. Whatever stays in this capsular space is going to go along the proximal convoluted tubule, which is here. Proximal convoluted tubule, that tubular fluid, so here it's called filtrate, and then once it enters the proximal convoluted tubule, we call that tubular fluid. So that fluid is going through the PCT, the descending, ascending parts of the loop of Henle, and now we're in the DCT. From the DCT, those will then connect to these long structures called the collecting ducts. When we look at this model right here, this is going to be the PCT, or the proximal convoluted tubule. If you remember from AP1, what does proximal mean? Well, we always said it's closest to the point of attachment, but when you get into AP2, I teach you that we actually have other ways of using the words proximal and distal. So really, proximal is going to refer to closest to the point of reference. And what is our reference point here? Our reference in the kidney is going to be the renal corpuscle. So the part of the kidney tubule, this proximal convoluted tubule that is closest to the renal corpuscle, that is proximal. So that is why this is the PCT. If we were to continue following this, that goes into the loop of Henle or the nephron loop, and then we've got different parts of that. This would be descending, ascending, this would be thick segment, thin segment, and if you think back to AP1 when we were going over tissue types, why did you have to know things like simple cuboidal epithelium? Well, it's because that simple cuboidal epithelium is what makes up part of the kidney tubules. So you can guess as we move along after this ascending limb, this part of the tubule is the distal convoluted tubule. And if you're asking yourself, well, that's right next to that renal corpuscle, why is that distal? Remember, it's farthest away from the point of reference. So if I were to grab this piece of the tubule and pull it out, it would be way back here. It's the same reason, and I demonstrate this in my class where I ask my students, what is the most distal portion of my upper extremity? Well, it's going to be my fingertips because it's farthest away from the point of attachment, which would be my shoulder joint. Now, what if I put my fingertips on my shoulder? Does that make it proximal? No, my fingertips would still be the most distal structure. So the same thing here, even though this comes right up next to that renal corpuscle, this is the distal convoluted tubule, and it will be really important, again, when we start talking about the juxtaglomerular apparatus, why we understand that this is the DCT. Okay, so we've left the collecting duct. Remember that the collecting duct is going to run through the renal pyramids. So these are all the renal pyramids. Remind yourself that the renal pyramid is the structure, but what region is it in? It is within the region of the renal medulla. And actually these collecting ducts are what make the pyramids look stripy. You're seeing the manifestation of all those many collecting ducts there. From the collecting duct, that will open up into the tip of the pyramid, which is called the renal papilla. That papilla is then going to open up into this small passageway called the minor calyx. So calyx is the singular version of the plural word calyces. So there will be a minor calyx connected to each pyramid at that renal papilla. When that tubular fluid comes through what's called the papillary duct into this renal papilla and it enters this minor calyx, now we have urine. So this is kind of like when we talk about this in the digestive system, when does a food bolus become feces? Well, that's in the cecum, and here this is going to be where that fluid becomes urine. The minor calyces are going to come together and they will form a larger passageway called the major calyx. So the minors will turn into the majors. And then the majors, and they're about 2 to 3 for each kidney, those major calyces will open up into this larger area called the renal pelvis. From the renal pelvis, then that will form the ureter, and now we've officially, that urine has exited the kidney and we can start talking about the bladder. By the way, if you've ever had a kidney stone, that intense pain that comes with that stone leaving your body is when that stone has entered the ureter. So there isn't much pain associated with it when it's in that pelvis, but as soon as that thing starts trying to leave your body, that is when it hurts a whole lot. So let's go ahead and look at the bladder and make sure we understand the final structures for the urinary system. The urinary bladder is a retroperitoneal organ, and actually only its superior surface is covered by that parietal peritoneum. So just another little bridge back to A&P1. Here we have the right ureter, and this is the left ureter. And how do I know that's right and left? I'll point that out in a little bit here. So if we were to open this up, these openings are the ureteral orifices, or the ureteral openings. You can see this is where this comes into the urinary bladder here, this left one right there. This is the opening into the passageway called the urethra. And ureteral and urethral sound very similar, so that's why I try to really overemphasize these words when I'm teaching it, so you guys know exactly what I'm saying. But the top two corners of this imaginary triangle right here are those ureteral orifices. The bottom, or the tip of the triangle, is the urethral opening. Okay, so it's the start of that urethra. And that triangle area is what we call the trigone. The trigone is this kind of immovable part of the bladder that funnels urine. So all of these folds, or rugae, that allows the urinary bladder to distend or expand. So if you're like me and you've just been chugging this all day and then you've got to go, thank goodness we have rugae to allow our bladders to expand. We also have transitional epithelium that allows our bladders to distend as well. So from here, we're going to have the urethra. Now there are two sphincter muscles, very similar to what we saw in the digestive system. We have an internal and an external urethral sphincter. The internal urethral sphincter is going to be more superior. It's going to be near the tip of this pyramid. It is made of smooth muscle. So remind yourself, can we consciously control smooth muscle? We cannot. So when urine stimulates the muscle at that internal urethral sphincter, that's what gives us that urge, oh, I drank too much of this liquid goodness and now I've got to go potty. What makes it so that we hold our urine until we get to the bathroom, or I don't know, maybe you're on a road trip and so you're at the nearest tree? Well, those would be our external urethral sphincters. We don't really see it that well here, but just know on this model, it would be in this area right here. The external urethral sphincter is made up of skeletal muscle. And remember, we can consciously control skeletal muscle. So if there are any parents out there of little ones who are in the potty training phase, okay, this is what we're doing. We're trying to get our little ones to consciously become aware that they can control this external urethral sphincter until they make it to the potty. So again, remember the passageway is the urethra, but we have two sets of muscles. Remember, sphincter muscles are what allow the opening and closing of a part of the body to control the passageway of that. So another thing we want to ask is this a male or female bladder? Male and female bladders look similar, except for the external structures, the extra external structures that males have. And the thing that tells you immediately that this is a male bladder is this organ right here. This is the prostate gland, and males only have a prostate gland. We're going to talk about this gland again in the male reproductive system, but in males only, the urethra is going to run through that prostate gland, or part of the urethra. Remember, in a male, for reasons you can imagine, that urethra is actually going to keep going. And so we actually call this the prostatic urethra. It's given a special name because that is the part of the urethra that runs through the prostate. So make sure when you are looking at models, especially things that are nice and color-coded, that you're not just, walking by and you're just kind of looking at it. You're actually picking it up and you're really examining it. These structures that are on the posterior side are the seminal vesicles. These are male reproductive structures that we'll look at in that chapter. And so that's also what told me that this is the right side and the left side. So remember, if you can see this purple prostate, that is a male bladder. If this is not here, then you're looking at a female bladder. And of course, females will not have these seminal vesicles as well.
Want to stay up to date on my latest videos? Please hit like and subscribe. And don't forget to check my Instagram page at The Anatomy Gal. See you next time.